Hi guys, Kenna here. We're going to go ahead and visit cell division today and talk a little bit about development of organisms. So our main focuses for today are going to be to go over the process of cell division and the stages as we go ahead and identify how cell division occurs. Now, essential questions for today. Why do cells divide? So first and foremost, we need to understand, what role cell division plays in our lives and what role cell division plays in terms of solving problems for the cells themselves. We also need to understand what the role of the placenta is in terms of division, in terms of maintaining homeostasis and providing those life functions or characteristics we talked about at the beginning of the semester to the developing organism. We also want to get some idea of what apoptosis is and how that may seem originally to be counterproductive in terms of survival of the organism, but plays a really important role in terms of the function of animals in particular. What are the stages of mitosis? And this is probably going to be the bulk of today's activity in terms of identifying the different stages of mitosis and how long they occur and what events occur in each one of them. What is the role of the spindle in cell division? We'll focus on the spindle because it does a lot in terms of allowing the different stages to occur. And then lastly, how are mitosis and cytokinesis both similar and different? Now, before we actually go into the nitty gritty of all these different pieces of the processes, the origin of everything kind of begins in a way that's pretty spectacular. We all begin experience existence as the simplest thing you can possibly imagine, if you're trying to picture life at least. A single cell, nothing but a tiny spherical bag of proteins. In fact, the average one celled amoeba looks far perkier under a microscope than a fertilized human egg. Yet that humble and nondescript appearance is highly deceptive. The egg, after all, boasts dazzling prospects, a future of truly dizzying complexity. When an amoeba divides, all you get is, well, more amoebas. But this is where the magic happens with multicellular animals. That single cell begins to divide, first into two, then four, then 8, 16, 32, 64, and so on. It embarks on a journey that will, over the next 9 months, produce cells with a huge variety of shapes and functions: bone cells, nerve cells, red and white blood cells, the cells of the eyes, the fingernails, the stomach, the skin. These early divisions are known as cleavage. Cleavage is the rapid succession of cell divisions that produces a ball of cells, a multicellular embryo from the zygote. Okay? The zygote continues to travel down the fallopian tube, forming a blastocyst. The blastocyst forms a fluid-filled cavity in the center of a ball of cells. There are three layers to the blastocyst, first of which is the inner cell mass, which you can see right here. Then we've got the trophoblast, which is this outer section here. And then you've got the blastocoel, which is this kind of fluid-filled center here in the middle. This image also includes the zona pellucida, which I'm not going to go ahead and discuss in this particular video. Now, these specialized different tissues will begin the process of specialization as the cell mass moves further down the fallopian tube. It is this blastocyst that implants into the maternal endometrium to go ahead and allow the baby to develop. Okay? As we start looking at this, these early embryo cells are what we consider to be totipotent. That means they have the ability to become any different tissue in the entire body. But as development continues, as we start moving along and getting those three different layers, the cells become determined. They now have a specific fate. Okay? In fact, the further along in its development a cell is, the less able a cell is to change its fate. A cell can be taken from one area and placed into another in earlier stages and changes its fate, but it only lasts for a certain amount of time. This development and differentiation of cells is partly affected by environmental factors, including what we call induction. Induction is the process by which one cell causes its neighboring cell to take a very specific developmental path. It's like they're communicating. They know they're going to have to work together. So one of them tells them, well, we're headed this direction. This is what we're going to be. And so they start working together to become better at that particular task. If you've heard about stem cells, this is a great example of where they come into play. You even have stem cells inside your body right at this moment, but these are what are considered adult stem cells or late stage stem cells. They're very limited in what they can become. But the earlier you go back in time to that initial fertilization, the more different things that cell can become. Totipotent means it can become anything. Pluripotent means it can become most things. And these late stage stem cells that you have in your body can only become a small handful of different types of tissues. If we choose a blood stem cell, it can become red blood cells, white blood cells, but it can only become blood cells. If we pick, bone stem cells, it can only produce bone tissue. Okay. So they become fairly specific in what they're able to become. Which leads us to gastrulation. Now, gastrulation is a phase early in the embryonic development of most animals, during which the single layered blastula we were talking about before begins to reorganize into a three-layered structure known as the gastrula. In humans, this takes place at about day 15. The three layers are labeled the ectoderm, endoderm, and mesoderm. And if you look at those prefixes, we can figure out where they are in that gastrula. Ecto means outer, endo means inner, and meso means middle. So this is in between the ecto and endoderm. So what kind of tissues can these become? Well, the gastrula layers develop into a wide variety of different tissues. The ectoderm is primarily going to be things that are on the outside. You got your epidermis of the skin, you got your epithelial lining of the mouth and rectum, you got your sense receptors that are going to be occurring in the epidermis, your cornea, the lens of your eye. But this is also going to help develop your nervous system, your adrenal medulla, tooth enamel. Okay. When we look at the mesoderm, this is what's going to develop the notochord, which will eventually develop into the spinal column, your skeletal system, muscular system, the muscular layers of the stomach, the intestine, the excretory system, circulatory and lymphatic systems, reproductive systems, except for the actual germ cells themselves, dermis of the skin, lining of the body cavity, and the adrenal cortex. The endoderm is going to produce the epithelial lining of the digestive tract, that of the respiratory system as well, lining of the urethra, urinary bladder, reproductive system. It'll produce the liver, the pancreas, the thymus, the thyroid, and the parathyroid glands. And that's just a kind of short list just to give you an idea of the way the tissues are going to develop. And as they begin to develop, there's one other term which at first seems a little backwards, but is really important for the development of animals in particular. Changes in the cell shape, its migration or movement to particular places, and programmed cell death give the developing animal its shape and form. We have talked a lot in this term about the relationship between form and function. The different shapes of the cells, of the tissues, of the organs helps them to be more efficient at doing their particular job or function. And we have a name for this idea of pre-programmed cell death, and it's called apoptosis. So you can think of apoptosis as kind of a timely, tidy suicide of cells. But I don't want you to think about it as just necessarily a bad thing, because this is what's absolutely necessary to create space between your fingers, between your toes, even between your legs. You may have heard of some of the developmental disorders where this doesn't happen properly. Maybe you've heard of something like mermaid syndrome, where they actually don't get proper apoptosis and the legs do not separate as the organism is developing. Okay. So, as the animal is developing and all of these things are happening, they need to go ahead and be fed. They need to bring in all their nutrients. They need to eliminate wastes, the cell transport things that we've already talked about. And how does the organism do that? Well, at least in mammals, this occurs through the placenta. So, as we look at the placenta during gestation, you have to recognize that it's right next to the maternal blood, but there's actually no direct mixture of blood between the offspring and the mother. Nutrients, gases, waste products are all passed through the placenta, but they don't actually share any blood. Unfortunately, however, this also means that things like drugs, alcohol, viruses can often pass through that placental wall as well. This also means that antibodies can pass through, and so that's important in terms of the development of the new organism. And this is because if we take a look and zoom in on the placenta, you've got these little circles or capillaries of blood exchange at these particular points. You'll notice that they are right next to the maternal ones, but they do not share any blood. Okay, so nutrients are going to go ahead and be pumped in through the maternal arteries. Those are then going to diffuse across through this thin barrier, which is going to allow them to be transferred into the fetal tissue and then be carried away by the fetal material. And you can see that this exchange is important for both organisms. And this is part of why it's so important to make sure both the baby and the mother are healthy. So, what exactly is the placenta? Well, it's interlocking fetal and maternal tissues that performs essentially the digestive, respiratory, and urinary functions for the fetus. Materials will be exchanged back and forth that separates across the membrane that separates the two bloodstreams. And if we take actual photos to look at, you can actually see some of these structures. So in this particular instance, you can go ahead and see the placental vessels and the placental cord. Okay, this is actually going off to the baby, and this is fairly early in its development. As time goes on, that still becomes more obvious. You can see the placenta up here and the umbilical cord that's going to connect this tissue from the mother to the developing embryo. Okay. Now, human development is just one example of this kind of mammalian development. And we divide it into 3 trimesters. Each is approximately 3 months long. It's not exact. And you can see the developmental stages here in these images as you go from an early embryo developing at about 5 weeks. Okay, notice as we look at the leg buds in particular, you still have the structure that will develop into a tail in other organisms. And you can see that the fingers look like they're in gloves. They're not separated yet at this particular point in time. However, as you get to 14 weeks, notice that the digits have started to separate. Apoptosis has happened appropriately. You're starting to lose any of the structures that would have developed into a tail. Spinal column has started to develop. The head has started rounding over as the skull begins to develop. Okay, this is now what we would typically call a fetus, no longer an embryo. And then by the end of the second trimester, about 20 weeks, the fetus grows about 30 centimeters in length. Now it looks much more like what we would identify as a traditional human. But all of this is amazing and magical. But why couldn't we just be one large cell? Why is it that we have to go through all these cellular divisions of cleavage, going to the blastula, going to the gastrula, and eventually developing into the organism that you see when you look in the mirror? Well, the problem is that the larger a cell becomes, the more demands are placed on that cell and its DNA. It's going to struggle to go ahead and move enough nutrients in and wastes out across the membrane. This is because as the length of a cell increases, its volume increases faster than its surface area. Okay, I might highlight that for you. Okay, that's a key concept we need to make sure that we have. Okay, its volume increases faster than its surface area. That's a key concept related to the idea of limiting cell growth. If you get too big, the cell begins to become self-toxic with wastes and begins to starve. So we don't want that to happen, and so the body solves that problem by going ahead and dividing. Okay. So here's just a quick example so you can kind of see what we're talking about. Now, when we start looking at animal cells in particular, they're not very square like this, but by using a nice square, we can go ahead and get the idea of how materials move into and out of a cellular structure. So if we were to have a cell that was 1 centimeter cubed, you can see that its surface area is 6 square centimeters. Compare that to its volume, which is 1 cubic centimeter. So that's a 6-to-1 ratio, which is decently favorable. However, as you go ahead and increase that to 2 centimeters per side, you are now looking at a surface area of 24 square centimeters and a volume of 8 cubic centimeters. 2 times 2 times 2. This gives you a ratio of 24 to 8, or a 3-to-1 ratio. So there is less surface area to the volume. If you do the same thing even more, going up to 3 by 3 by 3, the same type of pattern occurs. When you take a look at this, you can see 3 centimeters on each side gives you a surface area of 54 square centimeters. This is compared to a volume of 3 times 3 times 3, or 27 cubic centimeters. 54 to 27 is a 2-to-1 ratio. So the bigger the cell gets, the more difficulty it's going to have to get enough nutrition in and waste out. Your body solves this process by forming what are called 2 daughter cells. Now, I'm not exactly sure why they chose the term daughter, but they have here. And so we do this whenever we talk about cell division, even when we go ahead and talk about DNA replication. We're going to use the term daughter. The process by which a cell divides into 2 new daughter cells is called cell division. Now, cell division is part of the cell cycle. And the cell cycle represents the recurring events that take place in the period of time from the beginning of one cell division to the beginning of the next. In addition to cell division, the cell cycle includes periods when the cell is growing and actively producing materials it needs for the next division. So take a minute and think about, why is the cell cycle called a cycle at all? Okay, pause the video, see if you can answer this. So what did you decide? Hopefully you recognize that it's a cycle because it repeats itself. The same steps occur in order again and again and again. Okay, question number 2. Why do you think it's important for a cell to grow in size during the cell cycle? Pause the video here and see if you can answer it. So what did you say? Well, if you were saying that a division, if we just take one cell and keep dividing it in half, it's going to get smaller and smaller and smaller. So you need to grow if we're going to go ahead and keep the ultimate cell size approximately the same. Okay. All right. Now, question number 3. What might happen to a cell if all events leading up to the division took place as they should, but then the cell didn't divide? Pause the video and see if you can come up with an answer for this. So what did you think of? You're right if you said this would cause a problem. Okay. If the cell continues to get bigger, it still runs into that surface area to volume ratio issue that we talked about before. And this actually can be a problem that does occur with certain diseases like cancer. And so we'll talk about that a little bit today, but we'll focus more on cancer itself next class when we go ahead and start going over it. All right. So what are the parts of the cell cycle? Well, initially, we're going to divide it into 2 major subdivisions: interphase, what we typically refer to as the resting phase, and M phase, or mitosis, which the actual division occurs during. Now, calling interphase a resting phase is actually not a very accurate definition. There's a lot that occurs during interphase. It's just not the actual division of the cell. So when we go ahead and start looking at this and at M phase, let's go over the different stages. So during interphase, we have 3 parts. We have G1, S, and G2. During G1, the cell is going to grow. Remember, we said that's important to maintain cell size. In the S phase, we're going to replicate the DNA so that we can, when we divide, have DNA in both daughter cells. And in G2 phase, we're going to prepare ourselves for the actual division that's going to occur during M phase. In M phase, we're going to have the stages of mitosis. The stages of mitosis are prophase, metaphase, anaphase, and telophase. And we will talk a lot about what happens in each of those as we continue on with today's notes. There's one more part of M phase that's not technically in mitosis, but very closely related. And we'll talk about this later as well, and that's cytokinesis. Okay? Okay, so let's go back to the cell cycle. Now, during the cell cycle, cell's going to grow, okay, prepare for division, and divide into two daughter cells, each of which then is going to start over and do this whole process again. Again, that's why we call it a cycle. So the four phases of the cell cycle are G1, where the cell grows, S phase, where we have DNA replication occurring, G2, where we prepare for mitosis, and then M phase, which is going to include mitosis and cytokinesis. Here are those same stages, this time in graphic form, so that we can actually see what happens for each. So we have the G1 phase. So as you go ahead and look at the cell cycle, you'll notice that G1 says cell growth, S says DNA replication, and G2 says prep for mitosis. Now all three of these, G1, S, and G2, are part of the interphase. Once you've finished G2, then you enter the M phase. This is where actual cell division takes place, and there are two parts to cell division. The first is mitosis, prophase, metaphase, anaphase, and telophase, and then you see cytokinesis there at the end. Notice the cytokinesis wedge actually goes ahead and overlaps onto anaphase. So it starts at the same time in anaphase and finishes at the end of telophase. Then you'll notice that it goes back into G1 as the cell starts the cycle over again. Now, how much time does it spend in each one of these different phases? So as we go ahead and take a look at this, if the cell cycle is made up of 24 hours in total, one full day, 10 of those hours would be spent in G1. Remember that during G1, the cell is going to grow. Then you'd spend about 9 hours in the S phase. This is the time it takes to go ahead and replicate the DNA in the chromosomes. Then we have G2, which is the time it goes ahead and makes copies of the centrioles if it's an animal cell and prepares for mitosis. Only 1 hour out of that 24-hour time period is actually spent in M phase. So prophase, metaphase, anaphase, telophase, cytokinesis, all of this occurs in 1/24th of the life of the cell. So these are not very long stages. If we were in the classroom, you'd go ahead and see that as we go through a lab of the onion root tip, you're going to find far more cells that are in interphase, G1, S, and G2, than you are going to find in the different stages of mitosis, prophase, metaphase, anaphase, and telophase. So keep that in mind. All right, so the events of mitosis are divided into four primary phases: prophase, metaphase, anaphase, and telophase. And these steps enable the body to continuously make new body tissue for growth and repair. So that means that you guys, being younger, are going to do far more mitosis than somebody of my age who's kind of done growing. The only time we do any more mitosis is really to try and repair what's already there. So if we go ahead and take a look at these stages, you'll notice that up at the beginning here, okay, let's get our drawing tool out. We start out with prophase. Now, prophase, when we go ahead and look at it, we're going to go ahead and take the nucleolus right here, okay, and the nuclear membrane right here, and we're going to dissolve them. At the same time, we're going to go ahead and begin to condense or wind up the chromatin into chromosomes, which are the X's that we're more used to seeing. Okay, you can see here's the nuclear membrane beginning to dissolve. You don't even see the nucleolus anymore. And you can see we're starting to form X's here in terms of the chromosomes. Then, once that's complete, we're going to go ahead and see, okay, metaphase is where we line those X's up in the center of the cell on the center plate, which should make some sense because metaphase, meta means middle. Okay. Then you get to anaphase. Now, in anaphase, we're going to break apart the chromosomes and take them to opposite sides to allow for division. As we get into telophase, we're going to finish moving those chromatids to opposite sides of the cell, and we're going to start to divide and reform the nuclear membrane and the nucleolus. So when we actually talk about mitosis proper, what we're really focusing on is division of the nucleus. Okay. You see that after telophase, it goes back into interphase and then starts the whole thing over again. But really, what we're focusing on here is division of the nucleus. Now, this diagram is obviously fairly simplified, but it gives you the basic idea of what happens as we're going from a single cell to the two daughter cells in terms of the nucleus and the chromosomes. This is only possible because of the mitotic spindle. Okay. Now, when we go ahead and take a look at the spindle, this is a web type structure made up of microtubule fibers. So here's just another function of proteins. But it's essential for mitosis because it arranges the chromosomes into their correct positions for each of the stages to allow for optimal and efficient cell division. When we go ahead and do this, this is how we're able to move the chromosomes from one side of the cell to the other side of the cell to allow for the division into two parts. Okay, so when we start looking at the spindle, in metaphase, you'll notice that we have all the chromosomes lined up in the center. That's only possible because of these little lines you see here. And those little lines represent the mitotic spindle. In this particular case, they are attached to the mitotic center. In an animal cell, that would be the centrioles. But in a plant cell, there are no centrioles, and so that's going to attach to the mitotic center, which is next to the cell wall. Okay. So here's another image of it. In this case, it's obviously an animal cell because it has the centriole pair. And you notice that we have these microtubule fibers of protein coming off the centriole pair, and it's grabbing a hold of this little structure that holds the two chromatids together. So if these are the two chromatids of the pair, this is a centriole, okay? And that's where the actual fibers of the mitotic spindle will go ahead and attach. Okay. So let's go ahead and take a look at the different stages. So let's go ahead and begin with interphase. So remember, interphase is the longest of the phases, and this is where we have G1, S, and G2. Do you guys remember what happens during phase G1? That's right. The cell grows. What about during phase S? Good. That's where DNA replication occurs. And then G2 is going to go ahead and allow us to prepare for mitosis. Now, as we go ahead and go into mitosis, you'll notice that on these slides, we're showing you pictures of what it would look like. Okay, now in this particular picture, you're noticing that the cell has a nucleus that's intact. So the nuclear membrane is obviously there. And so this is obviously an animal cell that's in interphase. As we move into prophase, however, the chromatin, or unraveled DNA, begins to condense into the little X's you're used to seeing. And as it does so, you have paired chromosomes because we had replication occur during S phase. Okay. Prophase is the first stage of mitosis and is really close and happens pretty quickly with respect to that and metaphase. By condensing the unraveled DNA into the helix, we can now see the two different arms of the chromosome. Okay. The chromatin begins to condense to form the chromosomes, and each chromosome has already replicated, which gives us the little X format. This could, however, not be true if a mutation has occurred, but we'll talk more about mutations next semester. By the time we finish prophase, our nuclear membrane is completely gone, and the nucleolus has disappeared. Okay, the centrioles, which were put together during G2, have moved to opposite ends of the nucleus, and the spindle is forming to attach to those centromeres of the chromosomes. Then, once they've attached, they're going to pull the chromosomes towards the center, and as it moves towards the center, we move into metaphase. Now, the big characteristic that we see in metaphase, as you can see here, is that all of the chromosomes are lined up in the very center of the cell. They've been pulled there by the microtubule fibers. There is no nuclear membrane. There is no nucleolus. The nucleus is gone at this point in time. Okay? So, once we've got them all lined up in the center, the next thing we need to do is to begin to break them apart and separate them to the two opposite sides of the cell. The pairs are going to travel to opposite ends of the spindle. So we have half of our DNA on one side, half of the DNA on the other, which should make sense because we replicated it during the S phase. And now we're ready to divide the rest of the cell. Our DNA has been separated. As we go into telophase, the two new nuclei are formed as we reform the nuclear membrane and the nucleolus, and we start to move towards the point where we'll have two new daughter cells. By the time we complete telophase and cytokinesis, we will completely have moved from a single starting cell to two daughter cells. The chromosomes then unwind, the nucleolus reforms, and the nuclear membrane reforms. Now, something I want to point out about cytokinesis. If you literally translate this, this means division of the cytoplasm. So mitosis is the splitting of the nucleus, and cytokinesis is the splitting of the cytoplasm. Cytokinesis usually begins during anaphase and finishes as soon as telophase is over. To help you remember these stages, we have an acronym for you, and this is IPMAT, IPMAT. Now, technically, interphase, or the I, is not part of mitosis, but it is part of the cell cycle. So interphase, or I, not part of mitosis, but it is part of the cell cycle. That's what has G1, S, and G2. PMAT, prophase, metaphase, anaphase, and telophase, are the stages of mitosis. So if we start looking at mitosis and cytokinesis, you go from interphase, which remember is where you don't see the actual chromosomes because we're not actively dividing, but a lot happens here. In G1, the cell grows, S, the cell's DNA replicates, and G2, where we prepare for the mitosis that's going to begin occurring in prophase. In prophase, you begin to break down the nuclear membrane, the nucleolus dissolves. The centrioles begin to move to opposite sides of the cell so that they can go ahead and produce the spindle fibers, which will go ahead and attach to the centrioles, or the centerpieces of the chromosomes. Once this has happened, they are pulled to the center of the chromosome in metaphase, to the center of the cell in metaphase, where they're all lined up down the middle. Once this has occurred, then they get separated to opposite sides of the cell in the stage of anaphase. So now you have the individual chromatids moving to opposite sides. Again, all of this work is being carried out by the spindle fibers. And once those chromatids make it to opposite sides, we reform the nuclear membrane and the nucleolus. And we're doing cytokinesis, which remember typically starts during anaphase. So that by the time we're finished with telophase, we can finish cytokinesis, which is going to divide the cytoplasm and everything in it. And that allows the whole cell to begin the whole thing over again, starting out with G1, S, and G2, the stages of interphase. Okay, so those are the main parts of mitosis, cytokinesis, and the cell cycle. I wanted to give you a very brief introduction to basic genetics. We'll do a far deeper analysis of this next term, but here's a little basics to get you started. Each cell in the human body contains two sets of 23 chromosomes, one set from your mom, one set from your dad. In mitosis, we are going to do identical replication. The idea or goal is to have an exact copy of what you started with in the previous cell, which should make some sense because all of us started out as one cell and now are trillions of cells, and by the time you're an adult, hundreds or tens of thousands of hundreds of trillions of cells, and all of those started from one. Okay, it has to do this exactly each time. If it doesn't, then you have a mutation which could cause problems, and we'll talk more about that next term. So each cell thereafter has to have the same genetic material if we're making exact copies, which makes sense. So we can do a DNA analysis from any cell in your body and get the same results. Reproductive cells, however, like sperm and egg, only have one set of chromosomes, which is going to combine to make the new person with different genetic material. So you get half of your chromosomes from your mom, half your chromosomes from your dad, which gives you a new combination that is neither exactly the same as your mom nor exactly the same as your dad. Okay, so that's our notes for today. If you have any questions, please don't hesitate to reach out. I hope you guys have a great rest of your day, and I'll talk to you soon. Take care. Bye bye.